Hey everyone, and welcome to this explainer. Look, if you're a CBSE Grade 12 student gearing up for those biology or biotechnology board exams, you have landed in exactly the right place. Today, we are breaking down the fascinating world of genetically modified crops. Specifically, we're going to zero in on two absolutely massive case studies, BT cotton and the flavor saver tomato. I promise you a crystal clear, totally exam-ready breakdown of these concepts, and we're going to do it by looking at completely relatable, everyday examples. So let's get into it. To really wrap our heads around the science, we first need to understand the real-world struggle. Picture this. You're walking through a bustling, sweaty Indian sabzi mandi on a super hot summer afternoon. You pick up a tomato, and man, it looks absolutely perfect. But you know the truth, right? Give it 2 days, and it's going to turn into a soft, leaking mess in your fridge. Now, put yourself in a farmer's shoes. You've literally spent months growing these crops, pouring your sweat into the soil, only to watch crates of them spoil in a matter of days before they even sell. You'd be standing there thinking, why does all my hard work rot so fast? It's incredibly frustrating. Okay, so here is our game plan for today. We'll start with the core agricultural problem, define what GM crops actually are, dive deep into the flavor saver tomato, tackle BT cotton, and then wrap up by comparing the two solutions.
All right, let's jump straight into part one, the agricultural problem. Think of these as dual tragedies for our farmers. On one side of the coin, you have post-harvest losses. That's your tomatoes rotting in the market before anyone can even buy them. On the flip side, you have pre-harvest attacks. Imagine standing in a beautiful cotton field right at peak season, and suddenly, boom, insects like bollworms attack. And trust me, these guys eat faster than a teenager at an all-you-can-eat buffet. Entire crops can literally be destroyed overnight. So what happens? Farmers are forced to spray heavy pesticides again and again. That costs a fortune, it degrades the soil, and a lot of the time, they still end up losing their yield anyway. It is a totally vicious cycle of chemical use and financial loss.
Which brings us to part two. What exactly are GM crops?
So how do we actually fix this mess? We'll enter genetic modification. This is simply the process of inserting or silencing specific genes inside an organism's DNA to solve a very targeted problem. Think of it like using microscopic molecular scissors. Scientists can literally go into a plant's DNA, make super precise edits, and either give the plant a branding ability or just turn off a problematic one. It's really important to get this. It's not about creating weird Frankenstein monsters. It's just about giving plants the exact biological tools they need to survive.
Let's dive into our first big example. Part 3, the flavor saver tomato. Let's zoom in here, kind of like molecular detectives. See, scientists didn't just accept that tomatoes are destined to rot quickly. They asked the real question, what is actually causing this softening? Well, they found out that the spoilage problem is driven by one specific, totally natural enzyme. As the tomato ripens, this enzyme wakes up, and its entire job is to literally break down the fruit's cell walls. As those cell walls crumble, the tomato rapidly goes soft, loses its structure, and starts to rot. So that enzyme right there, that was the exact target our scientists needed to stop. Okay, grab your pens and pay close attention to this next term, antisense technology. Write this down, underline it, highlight it, because this is a massive, high-yield keyword for your board exams. Antisense technology is a genetic engineering method used to literally silence a specific gene. By silencing that gene, you completely stop it from producing its usual protein or enzyme. So for our tomato, scientists use this tech to silence the exact gene responsible for producing that annoying cell wall breaking enzyme. So here is the brilliant simplicity of the whole process, broken down into 3 easy steps. Step 1, identify the specific gene breaking down the cell wall. Step 2, apply antisense technology to silence it. Step 3, enjoy the result. Because without that enzyme tearing down the walls, the tomatoes stay firm for way longer. This was a total game changer. It gave farmers so much more time to transport and sell their crops without taking those massive post-harvest financial hits. If you remember just one concept from this section, make it this. They were pressing pause on spoilage. The flavor saver was never about making giant, weird-looking tomatoes or injecting them with artificial flavors. It was simply about outsmarting the natural rotting clock by hitting pause on that specific enzymatic breakdown. And honestly, this isn't some brand new science fiction we're talking about. The flavor saver was actually rolled out way back in the 1990s as one of the very first commercial GM crops. Biotechnology has actually been stepping up and solving these major agricultural roadblocks for decades.
Moving right along to part 4, blot cotton.
We are officially leaving the tomato market and stepping right into a vulnerable cotton field. Remember, when faced with massive insect attacks, the old school answer was just spray more pesticides. But scientists finally asked a much bolder question. What if the plant could actually protect itself? What if we could completely change the narrative from a helpless farmer spraying dangerous, expensive chemicals to actually empowering the plant from the inside out?
Okay, here is the exact biological mechanism you're going to need to know for your exams. Step 1. Scientists identified a common soil bacterium called Bacillus thuringiensis. And by the way, that's exactly what the BT stands for. This bacterium naturally produces a protein that happens to be super toxic to certain insects. So, using those molecular scissors we talked about, scientists extracted that specific toxin gene. Step 2. They inserted that exact gene straight into the cotton plant's DNA. Step 3. Now, as the cotton plant grows, it just naturally produces this insect-killing protein entirely on its own.
Now, right about now, you might be wondering, wait, is that actually safe for us? Yes, absolutely. And here's the absolute genius of how this targets pests. When a bollworm takes a bite out of the cotton leaf, it's basically biting into a plant that bites back. The BT toxin activates inside the insect's digestive tract, completely destroys its gut, and kills the pest. But, and this is the crucial part, because human biology and our digestive systems are completely different from a bollworm's, that exact same protein remains totally inactive and perfectly harmless to humans. To really lock this in your memory for test day, just picture this analogy. The plant becomes its own security guard. By simply adding the genetic instructions for this one specific protein, the cotton plant doesn't need a farmer out there defending it with chemical sprays anymore. That defense system is literally built right into the leaves. Let's tie it all together in part 5, comparing the solutions. So let's stack these two side by side to see the big picture. The Flavor Saver tomato is a post-harvest solution designed to increase shelf life, and it pulls this off by silencing an existing gene using antisense technology. On the other hand, BT cotton is a pre-harvest solution designed for pest resistance, and it achieves this by inserting a completely new gene borrowed from a soil bacterium. So you've got two huge agricultural problems solved by two completely different molecular strategies. Seriously, take a mental screenshot of this table right now. If you get an exam question asking you to compare these two, this is the exact breakdown you want to use. Flavor Saver tackles rapid rotting using antisense technology to silence that cell wall enzyme. BT cotton tackles bollworm attacks by inserting a toxin gene from Bacillus thuringiensis. This table right here perfectly summarizes the core concepts and mechanisms you need to memorize. All right, let's bring it home. What are the absolute biggest takeaways from our explainer today? Number 1, GM crops aren't some random chaotic experiment. They are highly specific, targeted scientific solutions. Number 2, they dramatically reduce massive financial and crop losses for our farmers. Number 3, they significantly cut down our reliance on heavy, environmentally harmful chemical pesticides. And finally, they are quietly but actively shaping the very food on your plate and the fields that grow it. Biotechnology is a remarkably powerful tool we use every single day. I want to leave you with a quick thought to ponder before you go. We've managed to press pause on spoilage in tomatoes, and we've turned cotton plants into their own internal security guards. As GM technology continues to evolve at a rapid pace, what other massive, everyday farming problems do you think we could solve next, just by tweaking a single gene? The possibilities in biotechnology are honestly limitless. Thank you so much for hanging out with me on this explainer. Best of luck with your exam preparation, and keep questioning the science around you.